Hi everybody, Dr. Burgess here. This is the Combined Science Biology higher tier run through. More than likely your teachers will run through it in the lesson, but this is for you to go through on your own. So first of all, it's out of 70, and as you'll know, there were 3 topics from paper 1 and 1 topic from paper 2. So first of all, this first question, it tells you glucose and fructose are different sugars. Fructose has a much sweeter taste than glucose. Suggest 2 reasons why the drinks company uses fructose in the drink. Do not refer to sweetness. So any 2 from these, so less is needed, which is good if you are a drinks company. You don't want to use as much fructose because it has a much sweeter taste. So it's less needed and it's cheaper to produce. There's also less energy in the drink, so people are more likely to buy it. 1.2, describe how a student could test the drink for sugar. Give the colour of a positive result. So this is about the sugar, the food tests for required practicals. So you needed to mention both. Well, for 3 marks, add Benedict's, heat the solution, and then any of those colours would give a positive result. Notice they'll give a range of colours, so if you're not quite too sure, it's worth a guess. 1.3, again, how the drinks are tested for protein, give the colour of a positive result. So this case is 2 marks, so add burette, and then it can be either mauve, purple, lilac, or pink purple. Question 1.4 is a 6 marker. Now, 6 mark questions, you need to kind of break those down. If you didn't do very well on this, you need to kind of break it down into different parts. So let's just have a quick look. So it says describe how protein and fat are digested, and they want you to talk about the enzymes involved and where the enzymes are produced. So I would probably talk about protein first and then fat. So as you can see, there's lots of answers you can have here, but the main thing is in terms of getting the marks, you've got to mention both enzymes. For protein, which is protease, and for fats is lipase. It says for a level 2, answers must describe digestion of fat and proteins. So that's to get into the higher marks. You've got to mention both, linked to the correct type of enzyme for both. Some of you are getting a bit confused with which enzyme, but if you look, there's lots of answers you can have. So what are they broken down into and where are they produced? In the lipase question, you can see that you can talk about lipase breaking it down into fatty acids and glycerol, produced in the pancreas, small intestine, but you can mention the role of bile as well. Next question. So this is kind of an experimental question related to osmosis. They're talking about the following. So they're looking at the concentration of salt. Control variables, they put the same volume of solution Z in it. The independent variable was measuring the mass, so they measured the mass of each tube. And the dependent variable, the independent variable, the factor that you're changing in this case is the different concentration of salt solutions. So it's really important to read the initial information. If you didn't read that information, the next question you may have got wrong. So what was the independent variable? Well, we look back, the independent variable is a thing that was changed, so it must be the concentration of salt solution. The student dried the outside of each tube. Why was it important to dry the tubes? Well, they were measuring the mass, as it said in the question. So the water or the solution or the tubing would affect or would increase the mass, or results would not be valid. 2.3. Okay, so they want you to calculate X, so percentage change. In an exam, if you don't know how to do percentage change and they give you lots of other percentage changes, what you can do is look to see if you can find the right answer using the data they've already given you. Okay, so basically you need to know the change in mass divided by the start mass times 100. Notice it says give the answer to 1 decimal place to get the 3rd mark. So basically 0.96 divided by 15.54 times 100 gives you 6.17%. So 1 dp would be 6.2%. Some of you didn't show your workings. Please do show your workings, because if you use the correct calculation but didn't get the correct answer, you can get a mark for that. So it's important you do that. Next one, so this is drawing a graph. This is really good marks, 3 marks for drawing a graph, and they actually tell you only use 0.2 to 1 molar per dm cubed. Draw a line of best fit, so you look at the percentage change from, let me just see if this works. Yeah, that one down to that one. That one down to that one. Right, so let's have a look at the answers. Okay, so you would plot it on the graph, and then it did say draw a line of best fit. So you get 2 marks for plotting correctly and 1 mark for doing a line of best fit. It could be straight, could be curved, as long as it's going through most of those points. So it then says determine the concentration of salt in solution Z. Well, basically that would be where there is no change in mass. So you draw a line through the point where it's 0, where your line of best fit goes through. And in this case, it would be 0.68, but it really does depend on where your line of best fit was. Question 3, it actually tells you this is about communicable diseases. So right, everything I know about communicable diseases. It talks about measles, and it tells you it is communicable. Okay, and it says what type of microorganism causes measles. It is a virus. Vaccinations help reduce the spread of measles. Suggest 2 ways the spread of measles can be reduced. Do not refer to vaccinations, so you can't mention that. So any 2 from these. So isolation of people with measles. You could cover your nose or mouth when you cough or sneeze, or frequent hand washing. You can't have just good hygiene. That's not enough. It's not a qualified answer. It then says describe how the measles vaccine helps a person to become immune to the measles pathogen. They give you 4 marks to do this. So describe. So basically from your revision. So you should know this. There are any 4 from here. So first of all, what does the vaccine contain? It contains a dead or inactive form of the virus. The measles pathogen or virus. White blood cells produce antibodies. So these antibodies are specific to that measles virus. You could give then a reference to memory cells, saying that when you get secondary exposure to the measles, or you're affected by measles, your antibodies are produced faster. 3.4, norovirus is a type of virus. Explain how they cause illness. 2 marks here. Okay, so you got to mention 2 things. Well, viruses enter cells, and then cells are damaged or killed. You could say the cells are burst open. But notice it's 2 marks for doing that. So a lot of you said that viruses enter cells. But don't forget the fact that cells then will be damaged or killed. Next question about clinical trials, and it gives 3 reasons why clinical trials are needed. So check drugs are not toxic, so toxicity. Check efficacy. Some of you are using the word efficiency. I think you misspelled efficacy, or you didn't remember the correct word. If you can't remember the word, don't write efficiency next time. Write to see if the drug works. That will be completely fine. Also, the 3rd one, many of you didn't get the 3rd one. It's to determine the dosage, or you could say just to find how much is needed. Question 4. So this was homeostasis, and it says hormones are released from endocrine glands. Which gland releases hormones to control other glands? Well, I think we said the master gland, so the gland that has many kind of functions is the pituitary gland. 4.2, it tells you about adrenaline can increase blood glucose. What is the one other effect of adrenaline? Well, it does increase your blood flow to your muscles. 4.3, cells in the pancreas detect changes in blood glucose. What type of cell in the body detects change? Well, it's any kind of receptor. So you've got to say a receptor or sensory cell. You can't say nerve cell, that's too general. You've got to mention that it's receptor or sensory. Right, 4.4. So this is getting a bit more difficult. So they tell you of an experiment. So scientists investigated whether blood glucose concentrations affect reaction times. They only used people with type 1 diabetes. Why did they do that? Well, that's something we need to think about for this question in a minute. So people with type 1 were selected. Explain why would they do that? Well, you can see you're recording the person's reaction time when the vastly different concentrations. Well, the reason is because blood glucose will vary more in type 1 diabetics. Why, though, for the second mark, why would it? So explaining why that is the case. It's because less or no insulin is produced when blood glucose increases, or you could say less or no insulin is produced to decrease blood glucose. 4.5 tells you about control variables. They give you 3 control variables, suggest 2 other that should be used in this investigation. So remember that looking at blood glucose concentrations affect reaction times. So they would allow any of these. So prior familiarity with reaction time tests, so maybe you've done it before. The type of test, so you could mention some control variables in terms of if they used hand, do they use their dominant hand, the positioning of the ruler, those kinds of things. Also, you need to control the amount of insulin because they're type 1 diabetics, the amount of sleep as well, and maybe distractions. So it was important for the scientists to monitor the health of each person during the investigation. Well, they've got type 1 diabetes, so if you don't monitor it, it could be quite dangerous for the people involved. So as it says here, low or high blood glucose concentrations are dangerous or unsafe. This next question, it stumped a lot of you, but when we break it down, it's not as hard as it looks. So they tell you people who do not have diabetes usually have a blood glucose concentration in the range of 4 to 7.8. So let's look at the graph there. Right, so we're talking about that. So non-diabetics, okay, their reaction time, you can see, is pretty good compared to those that may have diabetes, probably do have diabetes. So describe how the result in figure 6 shows the importance of homeostasis. Well, the lowest reaction times are found in people who do not have diabetes. Homeostasis keeps the blood glucose in a narrow, small range, or you could say lower reaction times could be a survival advantage. So when we say lower reaction times, we mean faster. So you could say faster reaction times rather than lower reaction times or lowest. So the fastest is found in non-diabetics. All right, so it keeps the blood glucose into a narrow or small range. Quite a difficult question, but easier when you can see the answer. Okay, right. 5. This question is about microscopy. So this is cell biology, shows a bacteria. For 1 mark, you've got to say the type of microscope, okay, and reason. Well, it tells you it's bacteria, so you should know from your revision you cannot really, well, you can't see a bacterium with a light microscope. They're too small, so you're going to need an electron microscope, not an electric microscope, an electron. And the reason is that that has a higher magnification or higher resolution. That's all you needed to say. It's only 1 mark. Some of you are writing lots for the reason, which wasn't worth doing when you got the type of microscope wrong. So try to just do quick answers for a 1 mark question. 5.2, testing, you know the differences between prokaryotic and eukaryotic. You could have talked about how, well, mostly you should have talked about prokaryotic cells are different. Could mention how eukaryotic cells are different as well, so the converse to that. So any 3 from these. So a lot of you knew that it didn't have a nucleus, or you could say DNA is free in the cytoplasm, or the genetic material is free in the cytoplasm. Many of you knew that it does not have a mitochondria. It has a single loop of DNA or has a single strand of DNA. It has plasmids and is smaller. Those are the only answers they'll accept. Some of you mentioned about flagella and tails. In the AQA spec, they don't accept flagella. 5.3. This stumped a lot of my students, and we need to go through this because this was 5 marks. So it shows a special slide used to determine the size of a cell viewing a microscope. It tells you each small division on the scale is 1.5. It says calculate the magnification of the cell shown. Okay, so first of all, we need to measure that distance with our rulers. Okay, not count these. You need to measure it with your rulers, and then that will give you the image, and then you can divide it by the actual. You can then work out the actual. I'll show you how to do that in a minute. So magnification, image divided by actual. So basically it's 36 millimeters. If you got between 35 and 37, that's acceptable. The conversion, so you have to then convert the millimeters into micrometers. So 36 times 1,000 to get 36,000 micrometers. The real size of the cell, well, you need to count these. Will this work? There to there, okay, which is 30. All right, and then it tells you each division is 1.5. So you can times it by 1.5, so that's 45. Okay, so then the magnification is the image, so the image is 36,000 divided by 45, and that'll give you 800 magnification. Now you can get some other marks, so some of you got 1 mark for that. If you did a magnification but using a different image size that you got, you also got 1 mark for that. Okay, so it's worth having a go. Some of you did not write anything, or you just wrote your answer. Please do show your workings, because you can get marks for your workings. Okay, smoking increases risk of lung cancer. It tells you that figure 5 is a lung cell dividing, so it's dividing, and it tells you describe the process of the cell cycle. Well, what I remember about the cell cycle is basically where we have two new genetically identical cells being produced through the process of mitosis. Now, because it's nearer the end of the exam, the examiners will expect you to use the correct terminology, okay? And they will expect it to be pretty spot on. So if you don't know mitosis, this is worth noting down. So DNA replicates. So the DNA is replicating, or 2 copies of each chromosome form, and then you can see 1 set of chromosomes is pulled to each end of the cell. Okay, because if they replicate, there's 2 chromosomes joined together, and then so 1 set of chromosomes will go one end and one end, the other to the other, and therefore the nucleus will divide, and then the cytoplasm or cell membrane divides, and you got to mention 2 genetically identical cells form. Many of you were saying 2 identical cells form. That won't get you the mark. It's got to be genetically identical cells. I also did notice that some of you are using the terminology for mitosis. At GCSE, they won't give you any credit for knowing the PMAT keywords. You won't get any extra marks. They don't want to see that because that's A level. So I know some GCSE revision places and things on YouTube. I have seen other people producing videos showing PMAT. You don't need to know PMAT, and I would strongly advise you just put what each thing means. But I would note that down because it's useful. 6.2, lung cancer is malignant, 3 marks. So you've got to not only describe what we mean by malignant tumour, but you've got to explain it as well. So the first thing it doesn't tell you what a malignant tumour is. Well, it's cancerous, therefore you can't just say it's cancerous. Unfortunately, you've got to say it has uncontrolled cell division, and because of that, it grows and therefore invades neighbouring tissues or invades tissues nearby, and then the cells spread to different parts of the body or tissues in the blood, lymph, or metastasis or metastasis. I can never say it. Metastasis just means it is being spread. So you've got to mention all of those three things to get those 3 marks. Okay, this next one. So this is the alveoli or alveolus, which is one of many alveoli. Okay, from the lungs of 2 people. Alveolus from a person who does not smoke. So person A does not smoke. Person B does. So you've got to then, it says that they do the same exercise. Why is person B breathing much faster than person A? Okay, so you need to look at the clues. All right, there are clues related to that, and this is all about kind of factors that affect gas exchange. So the first thing is you need to get, the reason they're breathing faster is to get more oxygen into their body. Why is they're having to breathe faster? Well, basically the alveolus in a person B has a smaller surface area, okay, and therefore less oxygen gets into the blood and therefore less respiration. It's as simple as that. Some of you are giving me very convoluted answers. Note how simple it is. That will get you the marks. And finally, question 7. Right, so I think some of you looked at this and said, angina. I don't know anything about angina. We didn't talk about angina. Well, the question's not really about angina. It's about coronary heart disease. Okay, and coronary heart disease, we have discussed what that is. It tells you already in the question that it causes chest pain and tiredness. So therefore, if it's in the question, you can't put it in your answer. People with angina are sometimes treated with a drug called GTN. We've never mentioned that before, but it tells you what GTN does. It widens the coronary arteries. And you've got to think, well, why is that a good thing? So it says, again, 6-mark question, you need to split it up. So it says causes and symptoms of angina and how GTN reduces the symptoms of angina. So you've got to talk about what causes this angina. So what kind of non-communicable risk factors, for example. Then you've got to explain how you get the symptoms. So you explain what is angina in terms of the coronary coronary arteries, and then you've got to talk about treatments. Okay, so how does GTN reduce the symptom of angina? So if we look at it in kind of in order, so causes of angina. Note you cannot say chest pain and tiredness, it's already been said. So things like high cholesterol, lack of exercise, smoking, increasing age, and a family history of atherosclerosis or heart problems. Explanation, so this is the key thing, explaining it. So what happens? Well, fatty deposits in the coronary arteries, you get narrowing of the coronary arteries. Why does that affect? Well, it reduces blood to the heart, or it reduces blood, oxygen or glucose to the body, less oxygen or glucose to respiration, less energy released, anaerobic respiration, lactic acid formed, muscle fatigue. Obviously, you are not going to be writing all of those things, but to get full marks, you need a couple of causes, a couple of explanations, and a couple of treatments. Your teachers will talk about how it is marked. It's not just marked, you don't get 1 mark per thing. It's very much, you've got, as it says here, level 3, relevant points, factors, effects are identified, given in detail and logically linked to form a clear account. Okay, so your teachers will talk about how it is marked as well. Treatment, more blood to the heart, therefore more oxygen, and then increased respiration, because it does say With GTN, it widens the coronary arteries, so why is that a good thing? Okay, so that is it really. So I do hope you found that useful. If you've got any questions, you ask your teachers, please. And also, it's important to set yourself targets to try and improve. And I hope you found it useful. I'll see you soon.